Hello everyone, I am Dr. Naveshya from the Department of Crop Physiology, SR University. Today I am going to cover on crop growth and analysis. So this is the one most important topic in crop physiology. So why? Because if we observe any plant at the different interval of time, so how we consider one plant has grown. So to analyze that, we have different parameters because just observation is not enough. So to consider one plant has a grown, we have certain parameters. So that one we will going to discuss in this today class. So first I will cover regarding plant growth. Later, what are the parameters we can use to analyze the growth of the crop. Later we will discuss. So first, let me tell you what is the difference between the growth and development. So growth is not a uniform changes. It is changes according to stage of the crop. It is not a series of reactions. So growth, if leaf changes its size or any volume of the leaf or dry weight of the leaf change also we consider it as a growth, plant growth, or leaf growth, flower growth. So it is not a collective process, it is a very singular process. So development means it is a whole series of qualitative quantitative changes from germination to till senescence. From germination to senescence, what are the changes going to take place? All the changes collectively we call it as a development. This is the basic difference between growth and development. So coming to stages of cellular growth, cell division takes place either through mitosis or meiosis. So cell division increases the cell number. After that, cell enlargement. After division, all the cells will increase its size or area of the cell. And after that, cell differentiation. In this stage, the cell changes to specific function. So simply we considered, for example, in tissue culture also we have callus. Callus means it is undifferentiated mass of cells. After cell division, we will get a mass of cell. We can't tell its specific role. Once mass of cell attains, it will go for cell enlargement, and after it attains specific function. And coming to growth curve, so if we keep a graph of dry weight over a time period, we will get this S-shaped curve. This is the growth curve. It looks like S-shape. That's why we call it as a S-shaped curve or sigmoid growth curve. It has three stages. All the plant, it is universal. The growth has three stages. One is lag phase, the log phase, stationary phase. Lag phase means this is the initial stage. During germination or initial seedling, the growth of the plant is very slow. That one we call it as a lag phase. Log phase or exponential phase means suddenly after certain stages, the growth of the plant will rise very high. So for example, it will for germination it will take 6 to 7 days, depend on crop species. After within 20 or 25 days, almost it will complete its juvenile growth and it will enter to the reproductive growth in 45 days or 50 days seedlings or crop. So that's why the log phase means it is a sudden rise in the growth of the plant. We considered it as the log phase. Later, after attainment of certain stages, the growth it won't increase. All the materials, whatever stored in the vegetative plant, it will transferred to reproductive organs like seed setting, fruit growth. So that stage we call it as a stationary phase, or it is where growth stops. And later, finally, senescence or death stage. Majorly, three stages we call lag phase, log phase, stationary phase. After stationary phase, the growth will gradually decreases. Finally, it stops because of death. So how we measure the growth means one is length or area or volume, number of cells in bacteria, algae, yeast. These are the different ways to measure the growth. One thing we can take a length of the stem, length of the root, or area of the leaves, area of the fruit or size of the fruit, and then we can go for number of cells in algae and bacteria. Like this we can measure the growth of the any organisms. So what are the factors affecting the growth means all the external factors required for the plant growth and development will affect the growth. Without these factors, the plant won't grow. So for example, light, of course, light is the very important parameters for growth. Temperature, water, mineral nutrition. In last lecture also we discussed how important it is, and water and light in first lecture we discussed about photosynthesis. There it has very specific role for everything. These nutrients or water, temperature, light, everything required for plant growth. Coming to phases of the plant growth, initial stage before germination we have imbibition of the seed, and after that the seeds undergo for germination. Later it enters to the vegetative stage or juvenile period. After that reproductive growth. At the end senescence. Same, it comes under lag phase. These two are comes under log phase, and finally reproductive stage, almost stationary phase. Senescence means final death of the plant. So we will get the sigmoid curve or x-shaped curve if we go for graph between dry weight of the plant and over a period of time. So coming to growth analysis, analysis of the yield influencing factor of plant development or net photosynthesis. What do you mean by growth analysis means? So in the name itself is there, analyzing, measuring the growth of the plant. One plant has grown. How we declare that statement means after analyzing through certain parameters. For example, if I give you some plants and tell me the height of the plant means we will use the scale to measure the height. Like that we have certain formulas are there to measure the growth of the plant. So these parameters are leaf area index, leaf area ratio, leaf area duration, and specific leaf area, specific leaf weight, net assimilation rate, and crop growth rate, and relative growth rate. All these are the analyzing parameters, and let the end harvest index. So if you want to analyze the plant is grown or not, you want to know about that means you need to go for all these parameters. So to tell that plant has grown, because for growth is not a singular function, it is a complex characteristics of the plant. You can't tell just by analyzing leaf area or leaf area index, we can't declare the characteristics of the plant. That's why to identify any drought tolerant crop or any high yielding varieties, we need to go for all these parameters. So these parameters will tell about the characteristics of the plant or efficiency of the plant. So one by one we will discuss with some calculations. So what do you mean by leaf area index means? It is the ratio of leaf area to the ground area. How much biomass is accumulated or how much biomass covered over the ground area, we call it as a leaf area index. It is the ratio of total leaf area to the corresponding ground area. So how we will measure the leaf area means first you need to take the leaf of the plant, all the leaves of the plant, and later you need to measure the leaf area of all leaves, and later you need to measure the ground area. Ground area nothing but spacing of the plant. For example, in wheat we will follow 20 by 10 means that is the ground area. 200 centimeter square is the ground area of the wheat crop. So total leaf area and ground area we call it as a ratio of these two, leaf area index. Leaf area index unit is, it's a unitless because the total leaf area unit is centimeter square, and corresponding ground area also if we measure in centimeter square both cancel, so the leaf area index unit is unitless. So let's now solve one problem. For example, you measure the leaf area. That leaf is around 4.5 meter square, and ground area also you need to measure in meter square only. So 1.5 meter square. So how we will calculate leaf area index means you need to divide this leaf area by ground area. It will give the leaf area index like 3, 2, 4, 5. So what do you mean by this value? Meaning means 3 means the leaf surface is 3 times the ground area. 3 indicates that optimum canopy development. So based on the leaf development, we will know about the leaf area index. If the area of the leaf is more, then leaf area index value also high. So maximum leaf area index indicate that the maximum light interception and also maximum photosynthesis just by covering the ground area. So I hope you understood leaf area index. Just you need to know that. So leaf area index means total leaf area divided by corresponding ground area of the crop. So ground area nothing but spacing. Here it is a complex value or combined value 1.5 meter square. You can use based on spacing 10 by 20 or 30 by 50 centimeter square. You can use both unit in a centimeter square also. Both are same. You will get same value. So coming to leaf area ratio. So ratio between the area of the leaves to the total biomass. Here we compare the area of the leaves with ground area, that will give you leaf area index. So leaf area ratio means based on the biomass how much leaf area plant has shown. So that ratio is known as leaf area ratio. It's measured in centimeter square per gram. The unit of the leaf area ratio is centimeter square per gram. What it indicates means it indicates the leafiness of the plant. If more dry weight means the less the leaf area. All the assimilates it is used to accumulate the dry matter. So based on that we will measure the leaf area ratio. So it is the ratio between the leaf area of the plant and dry weight of the plant shoot. First, what you need to measure? First, you need to measure the leaf area using leaf area meter, and later you need to take entire leaf or stem separately weight. So after the dry weight of entire plant and after that leaf area. This ratio will give you the leaf area ratio. So let's solve one problem. For example, if any plant having 200 centimeter square is the area of the leaves and total dry weight is 50 gram means you need to take the ratio of the leaf area and dry weight, you will get 40 centimeter square per gram. So this is the leaf area ratio, shows the greater leafiness. If more value means the area of the leaf is more. Same like leaf area index, area of the leaf increases means leaf area ratio also increases. More leaf area means it is directly proportional to the photosynthetic capacity of the plant. The plant is maintaining higher leaf area or lower leaf area. The leaf area is wide or thin. You can measure using leaf area ratio and also leaf area index. And this another parameter is leaf area duration. How many days it maintain the healthy leaves. That one you can know about by using leaf area duration. So its unit is centimeter square per, not centimeter square into day. So here leaf area you will need to multiply with the duration of the green leaves. Means how many days it maintain the specific optimum leaf area will give you the leaf area duration. Either it maintain for 2 days or 3 days. Based on that leaf area duration will change. So what you need to do, you need to take the leaf area, not stem. Only you need to take the leaf area and measure the leaf area. And you need to see how many days it maintains same leaf area. Then you can calculate this leaf area duration. And units also highly important because in most of the exams they will directly ask the unit of the growth analysis parameter. So for example, if you have two leaf area 1 you will get 300 centimeter square, and at the end of 10 days you will come to know that leaf area increased to 500 centimeter square. Then what you need to do means you need to take its average. So average is around 400 centimeter square. So after taking average you need to multiply with the time or days. So then you will get 4000 centimeter square a day. So this one is leaf area duration. So if the duration is more means it will clearly indicate that, so that one is maintaining healthy leaves, that plant maintain healthy leaves over a long period of time. So higher leaf area index reflects longer maintenance of active leaf area or healthy leaves. Higher, and it directly contributes to the higher biomass, and it like this we need to interpret this result. If two varieties are there or any two genotype is there, you need to see that performance of both the genotypes, which one is better, which one showing higher value, we will interpret that that variety or that genotype having higher biomass or yield, have capacity of yield more. Next one is specific leaf area. This is the another parameter. We are going to measure the plant growth. So one is how we measure the specific leaf area means you need to take the leaf area. Another one is total dry weight. So first you need to measure the leaf area and later you need to dry those leaves. If you have both leaf area value and dry weight of the leaves, you can measure using growth using specific leaf area. You need two values here. So if you have both leaf area and also dry weight of the leaves, you can use specific leaf area to define the growth of the plant. It's not like mandatory to take all the 8 to 9 parameters to tell one plant has grown. So based on the what values you have, you can choose the parameters to analyze the growth. So here specific leaf area is there. So how we will analyze the specific leaf area? So the first you need to take the leaf area and then dry weight if you have means if you take the ratio you will get the specific leaf area. Here you can see how to remember this formula means in specific leaf area is there. So just first you need to write leaf area then divided by dry weight. So the unit of specific leaf area is centimeter square per gram. So what specific leaf area indicates? Thin leaf with rapid. Almost it indicates same like area of the leaves, thinness of the leaves it will indicate. Another one is similar to specific leaf area, it's reverse value. Specific leaf weight means you need to measure first, it's the ratio of total dry weight divided by total leaf area of the plant. First same value, if you have this area of the leaves and dry weight of the leaves, you can measure both specific leaf area and specific leaf weight. What it shows means specific leaf area you will get to know the area of the plant over its dry weight. For example, 2 gram of the plant, how much area it holds. So those kind of values you will get, those kind of interpretation you can make. So in specific leaf weight it will show the thickness of the leaves. If specific leaf weight is more, then you can clearly say that the leaf is thick, not thin. The dry matter of the leaves is very high, you can say that. So to measure both specific leaf area and specific leaf weight, you need area of the leaf and also area of dry weight of the leaf. So if you have the dry weight of the leaves and also area of the leaves, you can go for specific leaf weight. So how we will measure? It will measure the thickness and dense of the leaves. So specific leaf weight is also a ratio and its unit is gram per centimeter square. Here you can see dry weight. Here dry weight we measured in a gram and leaf area measured in centimeter square. So if you remember the formula, you can easily analyze the unit of the parameter. And next coming to net assimilation rate. So this net assimilation rate over the duration of the time. For example, in 30 days plants you have some values and 60 days plant you have dry weight and leaf area of the plant means you need to measure between 60 to 30, sorry, between 30 to 60 days how much grown the plant shows, how much growth plant holds means you need to measure means you can use the net assimilation rate. So it is a 1 by leaf area into dW by dt. dW by dt means uh, total dry weight. It is the difference between the total dry weight of the plant and also duration of the plant. For example, 60 days and 30 days means you need to take the difference between 60 to 30 days. Like that, difference between dry weight of 60 to 30 days you need to take. That will give the dW by dt. and total leaf area of the plant at the end of 60 days you need to measure. So this much leaf area, how much dry weight of the plant will give you the how much plant grown between 30 to 60 days, like that. For example, if you have T1, for example, you have 20 days reading. 20 days plant show 20 grams. In 30 days plant show 50 grams of dry weight. And leaf area during this period is 2000 centimeters square. It maintains around 10 days same leaf area. Almost same leaf area means you can measure the net assimilation rate. So how we will measure this? 1 by 2000 centimeters square. This W2 minus W1, T2 minus T1. If you do, you will get this value. So this unit also highly important in most of the competitive exams. They will directly ask the unit of net assimilation rate. How we will measure means unit of this W is grams and unit of time is days. Unit of leaf area is centimeter square. That's why gram, dry weight gram. So per centimeter square leaf area and time per day. If you remember this formula, you can easily analyze or easily remember this unit of the any plant. So next one is how we will interpret the net assimilation rate. So net photosynthetic efficiency per unit leaf area, and also it gives the value regarding efficient conversion of assimilate into biomass. So this much leaf area, how much dry weight is there. So clearly indicates you the source sinks relationship. Source means the one which produces the photoassimilates. Sinks means the one which utilizes the photoassimilates. So how much the dry weight partitioning over leaf area you can measure using net assimilation rate. Coming to crop growth rate, so you want to measure the crop growth rate means we will use here ground area. So here we use leaf area, here we are going to use the ground area of the plant. So 1 by ground area is nothing but spacing, same dW by dt. dW means total dry weight of the plant shoot, dt means duration of the plant. So dry matter accumulated per unit land area gives the crop growth rate. So for to measure the crop growth rate, you need above ground crop sample like leaf, and also you need weight of the crop. You have both values and also you want to measure over certain time period this, you can analyze the crop growth rate. So unit of the crop growth rate is gram per centimeter square per day. So dry weight is gram, unit of ground area is centimeter square, and time is per day. So if you know the formula, you can easily know the crop growth rate. And one more thing, net assimilation rate and crop growth rate unit is same, gram per centimeter square per day, but this centimeter square is on ground area or spacing for crop growth rate. It refers to leaf area in net assimilation rate. Don't confuse, same unit, but the centimeter square is different. Centimeter square here represents the ground area, here centimeter square represents the leaf area of the plant. So one problem regarding crop growth rate. So at the T1, that is 30 days, dry matter of the plant is 100 grams. At T2 50 days, dry matter of the plant is 300 grams, and ground area is spacing is 2 meters square. If you have these values, you can easily calculate W2 minus W1. That is 300 minus 100 divided by T2 minus T1. 50 minus 30, 1 by 2. That is ground area. If you do that, you will get the value 5 gram per meter square per day. Here I mentioned centimeter, here I mentioned meter, means you can use both. If you keep this value in a centimeter square means the unit is gram per centimeter square per day. If you are keeping in a meter ground area, then here in the unit you need to mention gram per meter square per day. Depend upon the ground area unit, here the unit will change. So how to interpret this value? increased biomass per unit land area per day, it indicates, and for more value, better crop productivity under field condition. Over this certain area, over 2 meters square area, this much plant efficiently showing the dry matter, like that it indicates. So 5 gram over meter square area over time period. This is the value of the dry matter. Coming to relative growth rate, so relative growth means it is a comparison, comparing how much plant showing growth over a existing biomass. So it is the comparison between the existing biomass. For example, in certain time interval, how much plant grown over existing biomass, it will give the relative growth rate. Like it is the comparison. To comparing the two masses, we can use relative growth rate. Here we used in crop growth ground area over dry matter. So net assimilation rate means it refers to leaf area, how much dry matter plant holds. That one will give you the net assimilation rate. Relative growth rate means dry matter, how much is difference is there over existing dry matter. That one we will give the relative growth rate. So what is the formula of relative growth rate means 1 by W dW by dt. But here you see 1 by W means it is the total shoot dry mass and total dry mass of the plant shoot and duration of the plant. Same. Instead of leaf area, instead of ground area, here dry matter of existing biomass is there. It's just a theoretical value. If you measuring the relative growth rate over a short period of time, like over a 4 or 5 days, there is no much changes in the leaf area or plant biomass. At that time, you can go for this formula because the growth is not takes place linearly. So that's why there is an exponential growth curve. We are already by the in this lecture we already discussed about growth takes a shaped curve. Means it shows exponential growth. It's not a linear growth. So that's why over a short period of time we can use this formula. Otherwise we need to measure like ln w2 minus ln w1 divided by t2 minus t1. This one will gives you the relative growth rate in a standard way for longer time period. So for short time period, theoretical formula is 1 by W dW by dt. For example, if you are measuring 60 days reading means at the end of 60 days, how much dry matter is there existing biomass, you need to keep here. And it is a changing biomass. W2 minus W1, T2 minus T1, you can write it down. So standard means this one over a long period. You want to measure between 10 days to 60 days, between 50 days interval of time, how much growth the plant showed means that one you can measure using this formula ln w2 minus ln w1 t2 minus t1. So using logbook, you can measure LN2, LN3 values, but in exam the logbook they won't allow. That's why you need to remember certain L values. L12, this is the standard values, it won't change. So for every L for every crop, LN2 value is 0.693, LN3 is 1.098, LN4 is 1.386, LN5 is 1.609, LN6 is 1.791, ln10 is 2.303. It is a fixed value. Ln values won't change. So if you have logbook, you can measure this also. But in exam, we don't have logbook. At least try to remember this ln2, ln3, ln10. Most of the time, using these three values, you can measure the almost most of the values we can calculate ln2, ln3 and ln10, ln5. So ln5 also you need to remember. Remaining we can measure. For example, this is for the short period also. I am using ln formula. T1 is 10 days, dry weight is 50 grams, T2 20 days, and dry weight at the end of 20 days is 100 grams. My suggestion is don't go for this direct formula. Use this ln formula to get a correct value, because the growth will take place exponentially, not the growth doesn't take place linearly. We don't find linear changes over a time period. We find exponential growth. That's why please use this formula to identify the relative growth rate. So here we have two values, 10 days reading 50 grams, 20 days reading 100 grams. So relative growth rate, how we will need to measure ln 100 W2 minus ln 50 W1 divided by T2 minus T1. So ln 100 value means this is around 10. ln 10 is 2.03. We have one more formula is there. You want to multiply ln n into n means ln n plus ln n. You can do that. I will explain in next slides. So ln 10 plus ln 10. If you do that, you will get the ln 100. So ln 10 is 2.303 plus 2.303. If you do, you will get this ln 100. Like this, if you know this value 2, 3, 5 and 10, you can make the we can find all other values. So ln 10, using ln 10 you can measure the ln 100. ln 50 how we will measure means, just you see ln 5 is 1.0, sorry 609, and ln 10 is 2.303. So ln 50 means 5 into 10. So you need to add these two values. 5 into 10 gives you 50. That's why 2.303 plus 1.609, if you add these two values you will get this 3.912. So in exam how to measure means, please try to remember the standard values of 4 ln2 ln3 ln5 and ln10. Then you can easily analyze the relative growth rate. So difference of the W2 minus W1 is 0.693. If you multiply it by interval, you will get this gram per day. Why the unit is gram per day? W2 also, for example, here I will explain. This W is also a gram, and 1 by W is also a gram. T is a day. Like that, gram, day. That's why unit is gram per day. So how to interpret? So it represents the growth per unit of existing biomass and also maximum during early growth stage. It will show the rate of the growth, and it will also show the efficiency of the growth, and it will also indicates the how much plant have capacity to maintain the dry weight over the existing biomass. You can clearly indicate using relative growth rate. So this is for your purpose. Some like this we have one rule is there, ln A by B is equal to ln A minus ln B. Otherwise ln A into B is equal to, if you dividing means minus will come, if you are multiplying it means plus will come. So here we are multiplying. So ln 100 means 10 into 10. 50 means 10 into 5. So that's why we need to add here ln A plus ln B. If you are dividing means, for example, you want to know about the ln 10 value means 100 by 10. So that time you need to measure, that time you need to use this formula ln A by ln A minus ln B. For example, you want to analyze ln4. Why I mentioned only 2, 3, 10 and 5 value means, for example, you want to measure this ln4. So ln4 we can write it as a 2 into 2. So ln4, ln2 multiply means addition. So ln2 plus ln2. So 0.63 69 plus 0.69, we will get this 1.386. So ln4 value is 1.386. That's why no need to remember ln4 and 4 and 6. So ln6 means when 2 into 3 will gives you the ln6. 2 value is 0.693. 3 value is 1.098. If you add, you will get this 6 value. So that's why please remember 2, 3, 5. There is no alteration you need to remember, because we can't split or we can't multiply this 5. So better you need to remember this ln 5. And ln 10 2.303, ln 100 4, Just add 10 plus 10 because you need to multiply means ln 10 plus ln 10 4.6 and 1000 means 3 times you need to multiply, you will get the 6.908. I hope you understood about these things. So if you know these values, you can easily calculate this relative growth rate. Relative growth rate, most of the time we follow this lnW2 minus lnW1 T2 minus T1 only for the short time or only for the direct values of existing biomass. Very 1 or 2 days you need to measure the relative growth rate. Means you can theoretical formula you can directly use. For example, you want to measure around 5 days difference. Fifth day means T2 minus T1 is 5 days and W2, W1. At the end of the 10th day, you need to measure existing biomass. You need to mention here. Then you can use this formula. No need of this LN, no need of logbook. Nothing is required. Just direct values you need to apply and analyze this relative growth rate. I think this one also I covered. At the end, harvest index is highly efficient because it will show the how much biomass, how much vegetative growth is converted into a reproductive growth. So it will directly shows the source sinks relationship, partition of photo assimilates. It indicates that's why in all the almost all research, how they will measure yield like that they will add one more parameters harvest index in their research parameters. So how we will measure this harvest index? Economic yield by biological yield will give you the harvest index value. So usually harvest index we can measure it as a ratio in the decimal also. Otherwise harvest index we can multiply by 100 and we can keep it in a percentage. So one problem is there, seed yield is 3500 kg per hectare, straw yield is 6500 kg per hectare. Intentionally I mentioned this straw yield instead of biological yield. Biological yield means total yield, seed plus vegetative growth. So economic yield means only the seed yield. That one we call it as the economic yield. Biological yield means overall yield plus vegetative growth plus yield, grain yield, we call it as a biological yield. So here what they mentioned, seed yield means 3500 kg per hectare. Straw yield, straw yield means only vegetative part yield is 6500 kg per hectare. That time to identify the biological yield, you need to add both seed yield plus straw yield. Then it around 10,000. After that, to find harvest index, you need to take the ratio of seed yield over biological yield. What is meaning of this means in this much yield, how much seed is there? So how much economic part is there? How efficiently vegetative growth is converted into a reproductive growth? So you will get it. So 35%. 35% of overall yield is the seed yield. Like that meaning. So 35% of total biomass is converted into a economic yield. Like that you need to interpret this harvest index value. So moderate harvest index means balanced vegetative and reproductive growth. 35 to 45% you will get in most of the crop. Less harvest index, it clearly indicates that more vegetative growth and less yield. Almost all the resource or photo assimilates the plant utilized for its vegetative growth, not for its reproductive growth. Like that, we can interpret the results. High harvest index means all the whatever the stored photo assimilates is there in the vegetative, it clearly transferred towards the seed setting. So clearly transferred to vegetative, sorry, reproductive yield. So it clearly indicates the partitioning of the dry matters. So improving harvest index directly enhance the crop yield efficiency. More harvest index means more yield. So that's why scientists focus on the harvest index more because it clearly shows the how much vegetative growth is there, how much reproductive growth is there, how much it transferred from biomass to seed yield. So that those parameters we can analyze using harvest index. I hope you get some knowledge regarding these growth parameters. So one time as a revision purpose just to see these things. So leaf area index means it's the ratio of leaf area by ground area. Leaf area ratio means it is the ratio of leaf area over dry weight, not ground area. Ground area means leaf area index, and leaf area ratio means it's a dry weight. So how many days it maintains the healthy leaves? That one we call it as a leaf area duration. Specific leaf area means it is the ratio of leaf area by dry weight, and specific leaf weight means it is the ratio of dry weight by leaf area. That leaf specific leaf area shows the area of the leaves. Specific leaf weight shows the thickness of the leaves. And coming to net assimilation rate, it also indicates the assimilating ratio over this leaf area, total leaf area, how much dry weight it's maintained. That one you can measure using net assimilation rate. And next one is crop growth rate. Crop growth rate, we use ground area to measure the crop growth rate. Relative growth rate means how much weight changes over existing biomass. So that one you can calculate using relative growth rate. We have two types. One is theoretical formula is 1 by W dW by dt, and standard formula is ln W2 minus ln W1 T2. To identify the relative growth, please remember ln2, ln3 and ln5 and ln10. I already mentioned here. So if you know these values, the remaining value you can identify. So as summary, crop growth is a irreversible increase in the size. So to consider any plant grown or not, if I give you any two plants at the 30 days and 60 days, identify how much plant grown between these two interval means you need to measure using net assimilation rate, crop growth rate or relative growth rate. Using all these formulas, you can measure in that interval how much plant grown. So these parameters and these growth analyzing formulas will help you to analyze how much plant grown, how much plant developed. So for single formula can't define entire characteristics of the plant. So you need to analyze entire growth means you need to use multiple characteristics or multiple parameters to consider one plant has a grown. Okay, I hope you got some information regarding growth analysis, and thank you so much.